Hi, welcome back to MKF. Now we are going to discussing about detection of coliforms or enumeration of coliforms, Faecal coliforms and Escherichia coli by using USFDA BAM method, applying most probable number method technique. The most probable number method is a statistical multi-step assay consisting of three phases. First one is presumptive phase, second one is confirmative phase, and third one is completed phase. So by using these three phases, we can determine the coliforms, Faecal coliforms and Escherichia coli. In this enumeration process, serial dilution of the sample are inoculated into a broth media. Analysts score the number of gas positive colonies from each tube, means lactose fermenting tubes. From that, the remaining two phases of the assay will be carried out, and then uses the combination of positive results in phase 1 and phase 2, phase 3 by using the statistical table to estimate the number of coliforms present in it. In 1892, Sardinger proposed the use of E.coli as an indicator of fecal contamination. This was based on the premise that E.coli is abundant in animal and human feces and not usually found in other niches. Coliform is not a taxonomic classification, but rather working definition used to describe the facultative anaerobic gram-negative bacteria that can ferment lactose to produce acid and gas within 48 hours at 35 degrees centigrade. In 1914, US Public Health Service Department has adopted enumeration of coliforms as a more convenient standard for sanitary significance. Although coliforms were easily detected, their association with fecal contamination was questionable because sometimes coliforms were detected in environmental samples. This led to the introduction of Faecal coliforms as an indicator organisms. Now let us go in details. Enumeration of coliforms, Faecal coliforms and Escherichia coli by using USFDA BAM method by MPN techniques. So enumeration, sampling technique, sampling quantity from lot as per FDA BAM, sampling quantity from lot. So collect 8 OZ samples. 8 OZ means 226.8 grams of sample at least from a lot. Subsample, you have to collect the subsample. If the sample is in frozen condition, then if you want to do the enumeration process or microbiological analysis process, then you have to thaw the sample aseptically in a refrigerator temperature for 16 to 18 hours. If rapid thawing is desired, so you have to keep the frozen sample in a water bath below 45 degrees centigrade for 15 minutes if rapid thawing is desired for analysis of the samples. So this is what the procedure. And analytical unit as per FDA BAM, so 50 grams of meat or shrimp samples we have to take and dissolve in 450 ml of phosphate buffer solution. So blend for 2 minutes in a stomacher blender. So this will give rise the 10 to the power of -1 dilution. 50 grams of shrimp sample dissolve in 450 ml of Butterfield phosphate buffer solution. This gives rise 10 to the power of -1. Then make as decimal dilutions up to 3 dilutions. So again take 10 ml of 10 to the power of -1 dilution and transfer to 90 ml of PBS solution. This gives rise to 10 to the power of 2 dilution. And again shake it 25 times and again pick 10 ml of 10 to the power of -2 dilution and transfer to 90 ml of PBS solution. This will give rise to 10 to the power of 3 dilution. So this is how we can this is how we can do the decimal dilutions. Minimum 3 dilutions are required for performing the coliforms test in MPN methods. So after that, what we have to do is take 9 LST tubes. So LST means larval tryptose broth. So LST contains lactose. The coliforms are lactose fermenting bacteria. So take 9 test tubes of LST broth. So 10 ml of LST broth. Each test tube is filled with 10 ml of LST broth with Durham tube. Durham tube we have to keep in inverted position. So sterilized media LST. So each test tube contains 10 ml of LST with Durham tube. Then we have to add 1 ml of inoculum from 10 to the power minus 1 dilution to 3 test tubes, and 10 to the power minus 2 dilution to 3 test tubes of LST, and 10 to the power minus 3 also transfer 1 ml of inoculum to 10 ml of LST into the 3 test tubes. So 10 to the power 1 3 test tubes LST, and 10 to the power 2 3 test tubes LST, 10 to the power 3 also 3, total 9 test tubes of LST is required, and incubate at 35 plus or minus 0.5 degrees centigrade. So this is called presumptive identification test. 24 hours plus or minus 2 hours we have to keep. If gas formation occurs in the Durham tube, if gas positive occurs in the Durham tube after 24 hours, then note and we'll go for the further steps, means confirmation test for coliforms we have to go. If no gas formation occurs in some tubes, so re-incubation may be required. So further we have to re-incubate 24 hours plus or minus 2 degrees centigrade, observe at 48 hours. Still no gas formation occurred, then discard the tubes. This is the presumptive identification test for using LST, larval tryptose broth, soya broth, which contains lactose. If gas positive, if gas occurred in gas formation observed in some test tubes, then we will go for the next step. So the next step is confirmation test for coliforms. This is the presumptive identification test for coliforms, Faecal coliforms, E.coli. Then we will go for confirmation test for coliforms. BGLB. So brilliant green lactose broth, BGLB. So perform confirm test on all presumptive positive tubes. In presumptive test LST, so 10 to the power 1 tube gets gas formation, and 10 to the power 2 1 tube gets gas formation, 10 to the power 3 no tubes gets gas formation. Then based on that we have to prepare the BGLB tubes, depends upon the dilution, respective dilutions. From each gas positive LST tubes, means 10 to the power 1 tube got gas positive, and 10 to the power 2 also 1 tube got gas positive, this one nothing, for example. Then transfer each gas positive LST tubes into the next step, means so from a positive LST tubes, transfer a loop full of suspension into BGLB tubes containing 5 ml of brilliant green lactose bile broth and inoculum. So incubate at 35 degrees centigrade plus or minus 5 degrees centigrade. This is the confirmation test for coliforms, and timing is 48 hours plus or minus 2 hours. We have to incubate in the incubator. So depends upon the positive results, we have to refer MPN table. So MPN per gram. So we can express the results coliforms MPN per gram. This is the coliforms confirmation test. And the next step is confirmation test for fecal coliforms. So from each gas positive tubes from presumptive test from LST tubes, transfer a loop full of inoculum to EC broth. From each dilution, from each gas positive tubes from LST, transfer to a loop full of culture to EC broth. EC broth, these tubes contains 5 ml of EC broth and loop full of inoculum. Incubate at 44.5 degrees centigrade. Incubate at 44.5 degrees centigrade. This is the confirmatory test for fecal coliforms. If gas formation won't occur after 24 hours of incubation, and re-incubate tubes for further period of 24 hours, means total 48 hours. So you observe the reading at 48 hours also. Even 48 hours also gas formation won't occur, then take it as a negative. Then whatever the cultures gives positive, So refer the MPN table. So if for example 10 to the power of 1, one tube gets gas positive, and 10 to the power of 2, one tube gets gas positive, and 10 to the power of 3, no tubes gets gas positive. So no bubbles were observed in Durham tube. So refer the MPN table and express the results for Faecal coliforms. So MPN per gram. Then we will go for completed test for E.coli by LEMB, levine, eosin, methylene blue, agar. So whatever the gas positive cultures, whatever the gas positive tubes observed in EC culture, then transfer the loop full of culture to the LEMB plates. If 10 to the power of 1, one tube gets gas positive, then collect the loop full, pick the loop full of culture and streak onto the 10 to the power of 1 plate. If 10 to the power of 2, one tube gets positive on EC broth, then transfer the loop full of culture to 10 to the power of 2 LEMB agar. Streak onto the LEMB agar and kept it for 35 degrees centigrade plus dark centered flat with or without metallic sheen. So that is the suspected colony characteristics. What are the suspected colonies observed in the plates? Refer MPN table and express the result as MPN per gram. So this is about the complete test for Escherichia coli, LEMB test. Then after this, you want to go to further test, then pick the suspected colonies from LEMB plate and streak onto a PCA, plate count agar slants. So PCA agar slants we have to streak onto plate count agar slants and kept for 24 hours incubation, 24 to 48 hours. Then after that you can do the IMBC test, indole test, methyl red test, Voges Prosker and acetyl test. E.coli indole test gives positive and methyl red test also will give positive, and gram staining also we have to do. Further test for biochemical test also we can carry out. So gram staining also E.coli is the gram negative bacteria. So that's all about the video. Thank you universe for giving me the opportunity to learn and share the knowledge to the needy. Thank you and